let's think about this citizen science perspective. So I kind of gave you the rundown of like why I care, right? I want people, in the most discrete terms, to take photographs that have scientific value. And so that is my primary goal. But as a society, right, we are engaged in some important service work. So our first contract that we've ever received as an organization. We are now engaged in this Region 2 development of the Rocky Mountain Native Plant Materials Program with the Forest Service and CNHP. So we have a multi-year funding source to help develop a program that affects change in our restoration environment, in public lands, and what we specifically do is collect native seeds. And these seeds, they get sent to a nursery and they are increased. So our wild collected seeds are increased and then brought back to our districts where they are used in restoration projects. And so in year one, which was 2023, we like thought of ways that CONIPs could really interact and how we can facilitate this. But we're used to projects having fixed dates, right? And so the seed collection, it doesn't have a specific date because the seeds mature when they mature based on the weather. But what we found in the first year was that between CONIPs and the CNHP crew, we traveled 12,000 miles to scout for and collect seeds. And so the very first thing, why would we do something that we're talking about today and thinking about how to take more reliable photographs? To reduce the ecological footprint of the restoration project that we are now engaged in. Furthermore, it's a big place, right? Colorado is a huge state, and there's also Region 2 holdings in Kansas, South Dakota, Nebraska. And so we find that there's only so much time, right? CNHP has a couple of staff people, CONIPS has a couple of staff people and some volunteers, but we need your help to be better observers of plants so we can use our time wisely and get to those plant populations and collect those seeds for the maximum restoration benefit. And finally, why do we care? Why do we care? Because we feel like we need to do something. So I am a college professor, and I will tell you the number one prevailing feeling among my students is, what can we do? Like, what a mess, you know? How can we do something that makes a difference? And with your precious phone filled with all those rare metals, you can do something. You can take photos of native plants and build our information resource and help us find those seeds for restoration. And so we can all also be scientists. So this Region 2 Rocky Mountain Plant Materials Program I've been going on about is we assist in the effort to make native seeds part of the restoration regime in the United States of America. So you probably heard me give this other talk about how the word native wasn't really in any legislation since the beginning of public lands history in the United States. And so until very recently. So the idea that we would use native plant material for our restoration efforts is a pretty new idea. And so we want to honor that in our collective knowledge as a society is literally the greatest resource in terms of understanding and knowing our native plants so that we can affect this national effort, which is, by the way, funded by the infrastructure law, which you may have read about. So specifically, we want to find populations of selected species on Forest Service lands, and we want to collect those seeds. And maybe you won't interact with this project, but the process we're going to go through today is how we're doing this. So you can apply this knowledge in any way that you apply it, but if you want to help us find plants, this is a great way to do that. So in 2023, we looked for and collected seeds from these species. And if you look at the list, vast majority of these species are grasses. So if you think about it, region 2 is east of the Rocky Mountain front and all of Colorado. So that entire landscape is dominated by grasses. And so we think of these as our workforce species. They have really broad distribution, and they also have pretty reliable efficacy as restoration species in revegetation efforts. Okay, so I'm going to take a second to just take a breath, see if there's any questions. Please ask any questions in the chat. We're going to break the bindings, break the spines of our agrofields. But I want to get us all on the same page. So how are we going to use this dichotomous key, right? So as resource managers, we're just a society of lovely volunteers, but we work with a lot of land managers, and many of us are professionals in the environment, and we need to know plants, right? So we use the key to know the plants as best we can, and apply that measurement layer to our observational experience. So as you gaze upon this slide, if you have any questions, please put them in the chat.
I was wondering if you could start by defining what a dichotomous key is for people.
Yes, I'm going to do that right now.
Okay, great.
Yes. All right. So the very first step is to think about what this thing is. And so I thought the best way to look at that is thinking about the rubric for it, right? Like, what are the rules for building a dichotomous key? And so the first rule is that there's either or questions, right? There's yes or no questions. Dichotomous means two choices. Cotomy is like the act of making choices. And so when we think about what a dichotomous key is, it can take all sorts of forms. It can be really broad, like cover all the plants in the world or all the plants in the United States. For example, like North America, BONAP, the biota of North America. Or it can just be state specific or region specific, or it can be type of plant specific, right? So give me some reactions. Like I'm sure you guys have worked with the like the Wingate keys, right? You can get those from the Conib's bookstore. They're like $4 or $6 or something, and it's just a simple key of grasses. And so we jump right to POA, and it's furthermore only the grasses in Colorado. And then when we think about Ackerfield as a key, right? It's just the key of the flora of Colorado, but there are keys of every kind of flora, of every kind of fauna as well, right? So there are keys for insects and birds and reptiles and mammals and all sorts of stuff. When we use something like iNaturalist, that's not a key, right? But the data that it's employing in the in artificial intelligence world is related to this work in the key. So the dichotomous key is the primary tool that botanists use to identify plants. And as I was discussing before, like, why do we care, right? Well, we have to talk to each other. And so in history, human history, like the utility of plants was important, right? So communities of people might have their own names for identifying plants and connecting with them based on their utility or danger or whatever it might be. But as we grow as a global society, we want to use a single language. And it was Carl Linnaeus who was the first to really focus on this idea of developing a standard of two choices. And there's some Linnaeus was a very interesting human being. He was like obsessed with plant sex. So the first couple keys he put out were really about like sex parts. They were just like the plant sex parts. But there's so many exceptions to the rules that he created that over time, like that as a concept fell away. And it was it came to be not a valid concept. But it's just fun to learn about because you know I would say that he was dogmatically attached to a concept that like didn't have like scientific legs, but he eventually got there and started to build the taxonomic environment that we rely on today. And so we use binomial nomenclature to label species. And these words are Latin, and Latin is not a language that anyone speaks anymore, but a lot of our Romance languages are based on Latin and Greek roots. And so they are, in a sense, a universal language and word origin foundation that many people in the Western world can relate to. So in this binomial nomenclature, we think of plants as having two names, right? That they have like a genus and a species. But actually there's many layers of taxonomy that precede that. Like first, for example, we've got to get to plants in the great world of organisms. And then there's big categories of plant types, like for example, dichotomous plants or monocots or eudicots. And so we start to narrow it down. And so when we think about those names that we think of for plants, it's really the end of the road. And if you come to Carol Dawson's plant families talk, that's one of the interesting things about looking at the families is like nothing about the names that we employ like references to family. But that is actually one of the greatest like tools from a phenotypic perspective, right? Looking at how the families have similar characteristics. So in the dichotomous key, we eventually get to genus and species after reading through all the stuff before, which you might remember this from back in the day. Dumb King Philip came over for great spaghetti. Can I get some smiles on that one? So all the letters in the taxonomic environment might reflect the domain, the kingdom, the phylum, the class, the order, the family, the genus, and the species. So the name that we call plants is just the great spaghetti. It's not the stuff that came before. So let me just write this out. Hold on. So like we can usually work our way through the domain and kingdom real quick. We like we have more than one cell. We are multicellular, which makes us eukarya. And kingdom that we're discussing today is plantae. And then the phylum, class, order, that's kind of a lot of gray area. It's very interesting. It still relates to like phenotypic cursors that you can see. But we don't talk a lot about phylum, class, and order. But then family, genus, and species come into our active vernacular when we're working with plants. Okay, so rules for building a dichotomous key. The second rule we might laugh about in a few minutes. But we always give contrasting alternating characteristics in each couplet. But use clear cut opposites. Well, this is super hard in botany. And it really requires a deep understanding of the vocabulary. And so what might be contrasting to one mind might not be to others. Like, for example, I'm not sure how many words exactly there are for different kinds of hairs that can be on plants. But that is one that I've certainly stood on the trail and argued with people about. What it's hairy. Well, what kind of hair is that? Now thinking about the characteristics that are convenient and obvious features, this is a really important one for me, in thinking about the utility of what we're working on today. When like sometimes it might be unavoidable, and I think Ackerfield does a pretty good job of avoiding this, but like if the first character referenced in a dichotomous key is something you cannot see unless you have a microscope, it makes it a lot more difficult to carry out the work of choosing the couplets throughout the dichotomous key. But if you use characteristics that are visible, better yet, that are visible through all phonologies, like for example, the nature of the ligule on a grass, that makes it easier for identification. If, for example, you find that like one specimen that you're looking at for work or whatever, it like the first character is something specifically about the ovary, then you would have to wait until you could see the ovary, right? So you'd have to adopt this plant and check it out in different seasons. And when we think about keeping an eye on the species of interest, this is where iNaturalist becomes super helpful. I've used it a lot in like predictive, right? I'm supposed to run a statewide organization, but I can't be in the whole state at the entire time. So I can look at a species in a certain locale and I can learn a lot about its phenology by just like looking at the pictures that other people took. So that would help me to get to that plant at the right time for proper identification. And then the other thing, of course, is that we have analogies of ecology, right? So when I'm looking for a plant, right, I usually, if I'm really dialed into a plant, I start to get a sense for like the fact that it's on the northeast slope. It's always at like 8,500 feet, you know? And so you can start to link that with what you know about the plant identification, right? Is it in the right place? For your gut reaction to what identification like to make sense, right? If you're looking at like a mesic species, but you're on like a southwest slope, it's probably not that species. And so thinking about the ecological characteristics is helpful. All right. So every step further defining the dichotomous key is that you're going to make two choices, right? There's never like a trichotomous key where there's three choices. You have two choices. And so this is where our work will begin. Um, all right. So this might not be the way that you learned how to use the dichotomous key, but this is how I learned how to use one. And I think it's been pretty effective. And now I teach like a graduate level course and teach other people to do it. And hopefully that means that it's teaching people something. I will show you the work product of some of my students in just a minute. So to start this, I asked the question like, do you compose a song first or do you learn the song of another? Anyone here have children in fourth grade? When they get the recorder, no, it's most people's cameras are off. Hold on one second. Uh Yeah. So like you get an instrument, right? Not everybody, but some people might start just messing around with the instrument. You know, they don't try to play a song, but most people try to play a song. All right. So when you work from a known specimen, it's like going on a Conibs hike with some old timer who has a lot of experience, right? You can feel kind of confident in the observation and then you can suss out the details. And so it's just like knowing the song. And so my goal today is to make you familiar with how you might explore a plant that you want to know something about. Then you can take those skills, go on a hike, sit down next to a plant that you've never seen before, and maybe get somewhere with a key. And I already mentioned that using iNaturalist is helpful because you can access the great wide world of images therein. So when preparing for this talk, which I like just Googled like dichotomous key botany. And I urge you to do it. It's really funny. Because biology education, it is a language in itself, right? The key might be a language for plants, but generally we do the same thing to students all over the place as educators. And one thing we do is we make them create a dichotomous key. This is probably the only education that people get about a dichotomous key unless they enter a graduate program in botany. So I thought we could just spend a minute working on this. So I hope everyone is familiar with how to use the chat function, right? This is a classic biology lab. Create a dichotomous key with utensils from your kitchen. So remember that we got to think about couplets, right? So first we're going to do this exercise in the chat. So I'm going to first ask you about the fork, right? Write into the chat, like, what are some ways you can describe the fork? Fork. If you were just staring at a fork, what would you say about it? Okay, that's great. Okay, now Andrea England. Fork fits in your hand. That's a great one. But that will be somewhere up in like dumb King Philip, right? Came over. It's not in the Great Spaghetti because the fork fitting in your hand is going to define it as a utensil. Um, so it would be like the family of stuff. It's utensils. And then the fork, spoon, and knife are different like genuses within. So now let's try for spoon. What words would you use to describe a spoon? All right, and finally, let's get to a knife. Oh, and Annette, that's a great one. So like if we looked at the different serrations on a knife, right, that might be looking at leaf margins. And so we might define like what those serrations look like, and then ultimately decide that it's like a butter knife or steak knife or like bread knife or whatever it might be. All right. So now, if remember that by definition in the dichotomous key, we have to make one of we have to make choices, right? But it can only be two choices. So if you're up for the challenge, if we got to the family called utensils, all right, what might be the first couplet, the first either or yes or no question that will get us going down the road with fork, spoon, and knife? And remember, there's three of those things, but you have to make an either or choice. Oh, Sue, that's a great one. Is it one piece or separated? So, like, that one is super hard with plants because they might have a fused petal that then appears to have multiple petals, as you have to decide, but the petals are attached to each other. So it might be hard to decide if it's one piece or separated. All right. So we've gotten going here. We're thinking, right? So we'll finish this thought exercise. Hold on one second. If you're curious about this classic biology lab, you can just Google it and you'll find like a bunch of different examples of how you might continue to go on in this kind of practice. But I want to move on. Okay, so we got the idea, right, that we're gonna have to make either or yes or no choices, like it has this or it doesn't have this. But using just the dichotomous key alone, like I've never really been able to pull it off with just the key. The other things I need are the other book or reference of some kind. And so I focused on that plant identification terminology book. But just so you know, you can just like Google botanical terminology or like botanical glossary, and you'll get one. You could just print it out. Other thing you could do is, as I mentioned, I cut the glossary out of my Acker field, which I don't know. I mean, it's clever, but I often lose the glossary. So if I had it to do again, I would just photograph the glossary. I mean, take photocopies of it, make another copy. So I could have it in my hand. Um, the other thing that I find to be really helpful is to have post-it notes. So as I'm going through the key, I'm going to encounter words that I don't know the definition of. Sometimes I know what the definition is, but I look it up anyway, because I want to know like how Ackerfield defined that, right? And also I want to know specifically, like again, in the case of the hairs, the specific definition really matters. But the one that I have in my head that's like a generalization, I always want to confirm that. So I always keep post-it notes on hand and a pen of some kind. And I always bring post-it notes with me when I go on Conip's hikes. So those are things that you can use. You could also, of course, use index cards or a journal or whatever to curate your botanical vocabulary. Keep in mind that, like, in terms of learning, what does it mean to learn something? Writing and repetition are really important. So the more you can like write down those notes, right? Actually write them down, the more you'll commit this stuff to memory. And trust me, if you go through this activity like with a plant, you won't need those post-it notes again. But you can use that vocabulary base for other identifications. So the other thing I like about the post-it notes is like you can take the page off and then just like put it on the fridge or in the bathroom or wherever and continue to train yourself in practice with those words, right? To remember what they mean. If you're want to be in grad school or something and you want to be really self-involved, write the name on one side and the definition on the other, so you can use them like some sort of flashcard system. So sorry, struggling with my clicking of slides. Sorry about that. Oh, that was the last slide. So that's the end of that slideshow. Let me get myself organized. So, okay, Deb says having flash that's yes, vocabulary flashcards. I used to write this Spanish, on the post-it notes and put it all over the house. So Annette, I don't keep it in the place where I found the term in the book. I just keep it on a separate piece of paper so I can continue working the key and I can be looking at what those words mean and looking at the key at the same time. You can, of course, put the post-it note in the page of the identification you're working on, and that's helpful because then you can get back there really easily. Okay. Thanks. Always collecting tips. Yes. Sometimes when I'm doing the annotation, key annotation that we're going to work on now, I use the post-it notes. I actually cut these into like a bunch of skinny rows. So I can stick them on the page like as underlines under the choices I choose, but it's not good to mark up your key when you're identifying a plant, because then if you go to identify another one, the marking, you won't know which one it's addressing. So I don't recommend actually like writing in your key unless you are super focused on a certain species. Okay, let me pull this up. Okay. So I teach, as I mentioned, I teach an environmental studies department. A lot of my students are heading into careers in natural resource management, but they are not botanists. And so after the iNaturalist working group meetings, and thinking about like how we can get better at using the key, I started to employ this technique with my students because they are in a similar situation. They don't have like that deep theoretical knowledge. And so without further ado, we're going to get started. You can get your book out and prepare your space, because I will work on something and I will show it to you, but I'd love for you to follow along in the book. Um, So if you are looking at Ackerfield or any flora, somewhere on the inside cover is usually a map. And this map in Ackerfield is extremely important because she has these really cool maps that show you the geographic location of every species. So getting to know your county shapes is really helpful. So if you're looking in the inside cover of Ackerfield, you'll see a colorful map. I'm sorry, I did not think to make a slide of that. But it's just of the state of Colorado. So take a look and either use the book or use Google. Figure out where your county is. And if you can please write what county you're in the chat, that would be great. El Paso County has a nose. I never thought about that. Oh yeah, I guess it does. All right, super. So we're in that first place now. I want to navigate to one other place to show you one sec. So I keep going on and on about iNaturalist, but I haven't gone in here yet to show you. But the map on iNaturalist is like my favorite thing. So iNaturalist.org, and then you just open into this grid space. But then if you open into the map space, you can first be super impressed at how many people in the world are observing species. And then you can continue to drill down into our environment. And if you're doing this on your own computer, now go find your county, right? How active are folks in your county? How many observations are being made? Oh, I know what to make this easier. We can limit the search to just be plants and it won't be so overwhelming. So if you click up in this right corner on filters and you go to this little leaf, you'll just get the plant observations. Still pretty overwhelming. But cruise around and see how active your nearby community is. So interestingly, I was going on about the seed collection, but a lot of times we go in places where there's not a lot of people. So for example, when we go into like Pawnee Buttes or Pawnee National Grassland, going up north here, we see how there's fewer observations off of the highly populated areas to the east here. But then when we go in towards locations where people live and go hiking, there's gazillions of them. And actually, you can like find trails because see, once you drill down, you can see that all the observations are actually in a line. So that's a trail. And that's a fun way to go on a hike in the winter is pick a place where you want to go and go look at all the observations made there. Okay, so we have looked at the map in the front cover of Ackerfield and we thought about how we could get there in iNaturalist. And then on a day when you are having your cold, you want to take some time, I highly recommend that you read the introduction. It's really interesting. And it gives you a little bit of the history of botany in Colorado. And it also tells you about the major vegetation zones, and how to collect herbaria specimens, like how to make a plant collection, and importantly, to think about the life zone. So in Colorado, we have lots of life zones, and that's one of the coolest things about living here. So these life zones are related to elevation and aspect. And so from our lowest elevations, we have plains ecosystems, and then we go into the foothills and the montane and the subalpine and the alpine. And weaving through all of those life zones is the riparian. So when you have like a just regular field guide like that, where it just be like the trying to look on my shelf and think of one right now while I'm talking, but like a CMC guide, the wildflowers of the Front Range or something like that. You can use that in conjunction with the dichotomous key because it will tell you where that plant should be. And if you open your Acker field to any page, and look at any observation or any record, you'll see that there is an elevation range. So I just like open to Gaillardia pinnativida, and its range is 43 to 7,200 feet. So that tells me that like it would only be in the plains and the foothills. It couldn't possibly be on the top of Pikes Peak. Even if I thought that's what I was looking at, it's a process of elimination. So looking at those life zones is super helpful in the introduction. Okay. Then you are on page 20 in the second edition. And you start to go through a key to the families. And this is the part that's gonna really like knock your socks off. And we're not gonna do that, right? We're not keying families with Ackerfield. I urge you, like I said, to go to Carol's talk and figure out how to get to the family. And because I've literally never keyed a plant from key to families. So for example, why not? Well, there's lots of different kinds of plants. In the chat, could you name some general categories of plants, please? What kind of plants exist in the world? Okay, super. So in that entire list, as I suspected, all of those plants, except for seaweed and ferns, are actually all one kind of plant. Does anyone know what kind of plant they are? Yes, thank you. They are the angiosperms. So we can skip those first few pages and make a pretty general assumption that we're looking at an angiosperm, unless we are like in the ocean, or we're looking at a conifer, or we're looking like at a bryophyte, like a moss. And so I was like thinking about it. All the foods we eat are also angiosperms. The only one I could think of that wasn't was like anything that was on a conifer, like a pine nut. Can you think of any other examples? Like, I guess you could eat algae, like if you eat like spirulina. We obviously eat fungi, but fungi are not plants. They're not plants because they are they're heterotrophs. They require other organisms to help them to eat. Okay, so we're looking at angiosperms. And so when we get to that great place, we can start to make another giant, a giant classification break, right? There's two kinds of angiosperms we're familiar with. Does anyone know what they are? Two major groups. And their names relate to how they emerge, what the first seed leaves are like. Yes, thank you. So we have dicots and monocots, right? And so again, we don't need to use the key for this. We can generally figure out if a plant is monocot or a dicot without touching the key. So for example, I'll ask like, what are some characters of monocots that you guys can think of right off the top? Okay, great. So this one Annette has offered is a really important one that you can see on almost any plant. What are the veins like? So do the veins of the leaves all go in one direction or are they netted and go off into different directions? So we might think it's like flowers and grasses, but actually not a that's not a good way to think about it because there are monocots that look like flowers, like lilies, and grasses have flowers. So it's not really an actual division. So we've got the parallel veination. Another one is when they emerge as seedlings, all monocots have just one leaf. And that's why it's called a monocot. Um, because it has one collided monocot. And so if you are doing any winter sowing this season, you'll find that like your grasses will emerge as a single blade. But your dicots, on the other hand, they emerge as two seed leaves, two collidins. And so those early seed leaves are not very differentiated. They just look like round leaves. And then it's the next set of leaves, the first set of true leaves that have like the defining characteristics that you might find in the dichotomous key if the lead is asking about leaves. So we can make this other giant division, right, to move further into the key. We can figure out if we are trying to observe a monocot or a dicot, or in the case of trees, right, it could be a conifer. And we are going to actually start with a conifer. So let me get my page pulled up here. So when I teach this to my students, I tell them to be smart, right? You don't want to practice this with the most complex species right at the get-go. But the conifers are easier to start with because they are fewer in species number. So let's take a look here. If you go to the back of your book, right, you can let's start at Pinus. So that is on page 65. Okay, so we can start even with 64 just to go a little bit farther back. So we can start at the family, Pinaceae. And so when we first start there, we find that the pine family are evergreen trees. The leaves are linear. They're fascicled. The pollen cones are small, not woody, right? And so when we think about what is a pine tree, again, you can respond in the chat. What are the key defining characteristics for you? What is a pine tree? All right, well, that's a lot of good stuff. Good work. Um, okay, so if in the most simple terms, right, we would say that pine trees have needles, which are modified leaves, and they have cones. And the cones are kind of important because they also tell us that these are wind pollinated. Um, so we don't tend to find like these really fancy sex parts on these plants because they're not using their bodies to attract pollinators. They're using their bodies to like be in a position for the wind to carry the gametes. And then for the reproductive organ, the cone to catch that reproductive material in those woody bracts of the cone. So I like to start here because these keys are short. Um, so please humor me. Let's work on the ponderosa pine because this is one that I think we can all like, we know what it looks like, right? Close your eyes. You know what a ponderosa pine looks like. So thinking about known species, this one I feel like is known to everyone. Just giving you a little background about the special ponderosa pine. This is the largest uh, plant organism in the state of Colorado. It's the greatest biomass of any plant, and the ponderosa pine is widespread throughout the globe. But what I want you to think about is where it is, right? How does it get to be so big? It's pretty much nestled in that space where the slope goes from steep to mellow, right? So you're coming off the mountains and you're in that montane foothills, right where my two hands are meeting. So the when the slope is intense and steep, gravity wins, right? The water is pulling away from those habitats. And then when the slope is more mellow and flat, as in the foothills to the plains, we find that gravity wins again and the water goes into the ground rather than running off the landscape, right? So why are these giant trees where they are? They're in the place where the water stays still, right? All of that great runoff starts to sink into the soil when the slope of the landscape decreases. So the ponderosa pine, really important plant to our ecology and to indigenous peoples in Colorado for its many ethnobotanical uses. So now hold up your finger, right? Like another thing about the ponderosa pine is that it has the longest needles. And so you can easily identify that in the landscape because it has such long needles, but that relates to that ecology we were just talking about. It's a mesic type species compared to others. Now, when we think about the nature of the needle, right? That it is a needle. We skipped that, right? That was in the class, the phylum class order part of taxonomy. But just so you know, the needle as a concept is an adaptation for drought tolerance. Needles persist for 6 to 8 years, so it's not the same caloric investment that a deciduous leaf would be. And then so that's important, right? In these harsh landscapes that we live in where it's super windy and cold and all of that. And that needle persists for its photosynthetic value for a long time. So another interesting thing about our pines is that they come in pokey packets. So again, going back to early biology education, how can you tell the difference between a pine, a spruce, and a fir? Are some pretty simple little sayings. So we say that a pine has pokey packets. And so what that means is that the needles are all coming out of like a fascicle and you can count them. And counting them is part of that piece of measurement that we were talking about at the beginning when I was saying the difference between natural history and science is measurement, right? So Pines have pokey packets, just while we're there. Firs are flat and friendly. The needles actually only have two sides, and so if you hold a needle in your hand, it doesn't roll. And also you can like touch a branch of a fir and it's not particularly sharp. And then there are spiky square spruces. And spiky square spruces, as this implies, the needle you can roll in your finger because it has four sides, so it's square. Also, if you were to like touch a spruce, it's really pokey. So generally, again, these are just basic observational tools, right? To get to a place in the key where you're not starting from page one. So we're starting at pokey packets. We can all in our mind's eye imagine that. And then when you're holding your needle, your fingers up, another thing that we can really focus on is the length of those needles. So how many are there per fascicle and how long are they? And so the ponderosa pine leaves, the needles are the longest that you'll find in Colorado. They're like 4 to 5 inches long. And so that is another piece of measurement. So thinking about the utility in your flora, if you turn to the back page, there is a ruler. So that is super helpful to use because throughout the key, you're going to be asked about the length of things. Now I find that these length of things to be a real challenge to work with because the way plants grow really depends on moisture availability. Also, you might find in the couplet, remember we were talking earlier about how like it's supposed to be like clearly definitive dichotomous choices. Like if you get into acrofield, sometimes you find that like the range of length on a needle is overlapping with the range of length for another species. So you kind of have to rely on another character, like how many leaves per fascicle. So if you are in your Pinus ponderosa, I lost my page 65, right? This is one I love to work on because it's just so quick. So if you were in the field, right, we all pretty much agreed that you would be able to know that you were looking at a ponderosa or be fairly certain. So on page 65, if we look down under Pinus, we find Pinus ponderosa, variation scopulorum. And this is the magic of this process in this very simple species. So our key ends at 2A. Leaves mostly 9 to 20 centimeters long, usually 3 per fascicle, but sometimes 2. Okay, so if you're working with a known species, now what you have to do is go backwards. So how did we get to 2, right? We had to make a choice at 2. And so we go back to 1. In 1A, our choices were leaves are 2 to 3 per fascicle, go to number 2. Or 1B is leaves are 5 per fascicle, go to 4. And so you see, if we had chosen 5 or counted 5, we would skip the couplets 2 and 3 and go down to 4, but we don't need to do that right now. So then also taking a look at this half page, I just want to point out to you that as you're practicing this, like it's helpful to work in places where there are not that many different kinds of spaghetti, right? So in the Pinus family, there are 1, 2, 3, 4, 5, 6 species. But if you were to look up, like, for example, an astragalus or a penstemon, you would find that there's something on the order of 30 or 40 species. So that would be more difficult to work with, right? All right, so we made it through our first one, and this is hopefully you're like, wow, that was so easy. But we want to think about also the vocabulary. This one was easy because there was only one vocabulary word. Can someone write it in the chat? What is our only vocabulary word? Fascicle. Got it. All right, so if we turn to the glossary on page 818, we can learn that a fascicle is simply a cluster. Um, and then if I look in my plant ID terminology, it's a little harder to get there quickly in this one because you gotta go by, there's no like index. So let's see here. And if you look it up on the internet and you find a different definition, please share it. Oh, I'm not even finding fascicle that quickly in my plant ID terminology book, but it can be found. Page 45. Thank you, Sue. Okay, so Harris calls it a tight bundle or cluster. And also you can look here on to see a picture, but I'm going to show you a picture right here. Oops. All right, so right here, this is an eastern white pine, but that looks like pretty good approximation of a ponderosa needle. See how the needles are coming out of this little packet. So that's the pokey packet. Okay, so Sue, there's another term. I don't even know how to pronounce that one. Do you know how to pronounce that, Sue? Acicular. Is that what other people would say? Probably. That's why we use the Latin. It's not a language we speak. So that's a needle shape. So great one. And those are words you could write on your post-it notes so that you could remember them for the future. Oh, my apologies. I realized my screen share didn't switch over while I was blabbering on. There's the picture I was pulling up, the fascicle there. Okay. Ready for another? All right, so now let's look at pinyon pine. We're on the same page. And we're still on 65. Now, the pinyon pine, another interesting species. It is not our species with the largest biomass, but the pinyon pine does occupy like the largest geography across the globe. There are pinyon pines all over the world in similar latitudes. And so, like, for example, I was talking to Andrea England, who's in our group just yesterday, and she just went to Italy. And we were thinking, why are there pine nuts in pesto, basil pesto, food from Italy? Well, because there are pine trees there, or pinyon pine trees there. I assume they call them something slightly different, but I'm not exactly sure. So like, I don't know that the common name would be pinyon pine is what I'm trying to say, but Pinus edulis, yes. All right, so let's go back to this key. So we would, at our first choice, we would make the same choice, right? That there's 2 to 3 leaves per fascicle. But when we got to the second couplet, the first lead says leaves are mostly 9 to 20 centimeters long, but 2B says that the leaves are 1.5 to 9 centimeters long. And so if you can put a pinyon pine in your mind's eye and hold out your fingers to think about how long it is, right? A ponderosa pine needle is at least as long as your finger. But a pinyon pine needle would be about half your finger length. And so we get to 2B and then we go to 3. The trees are 15 meters tall with a twisted trunk. Scales of seed cones not prickly near the tip. Wingless seed cones. Okay, so here's this marker. So we saw that both the needles were shorter, but also the trees are shorter, right? Let's go back up here. There wasn't even any reference to how tall the ponderosa pine was in the key, but if you look in the species description, it says that the trees are 25 meters tall, so much taller. All right. And so the scales of the seed cones not prickly near the tip. Now that would be something that you could see all year long, right? And you can feel it. So what this means is that you can hold the cone in your hand and it doesn't like poke you. But like if you had like a bristlecone pine cone in your hand, it would hurt your hand. So that would be an example of there being prickers at the tip. So like, for example, the bristlecone pine I just mentioned falls out at 4A, and the leaves are 3 to 4 centimeters long, drops of resin, scales of seed cones with a bristle tip. And so that would hurt if you touched it. All right, so we got through 2 Pinus. Now, are there any questions right now? All right. So let's try for the Rocky Mountain juniper. So we're on page 63 now. And now we kind of skipped this part a bit. But we forgot to classify this idea of gymnosperms, right? So there's the angiosperms, the flowering plants, but I forgot to mention this term gymnosperms. And so our conifers are gymnosperms. And hold on, I gotta close my door one second. All right. And so when we are in the gymnosperms and we get to juniperus, juniper, right? We can go look at these couplets. So this is one that is actually not that easy to figure out with your observation skills on the trail. However, we in the Southeast discovered a great tool. So down here we have Rocky Mountain juniper and we have 1-seed juniper. And so it's actually really kind of hard to work the key to figure out the difference between Rocky Mountain juniper and 1-seed. But I just want to give you my anecdotal advice first. So in Native Plant Master 2 years ago, we figured out after looking at these that the Rocky Mountain juniper, if you take a branch and you lay it in your hand, it pretty much lays flat. It's pretty soft and the frond will lay flat in your hand. But the other very common juniper that we have is one seed, monosperma, and you cannot identify it by the number of seeds. That is a misnomer. But if you take a frond and you hold it in your pond hand, it stays up. It's like 3D. And so we came to call it 3D one seed. And then Rocky Mountain Juniper being the one that lays flat in your hand. And there's nothing about that in the key, but I just thought I would mention it. Okay, so when we are looking at the top of gymnosperms and thinking about the cypress family, we see this concept that the leaves are scale-like, right? We're no longer thinking about them being linear or needle-like. And so what we're observing is slightly different. So 1A, leaves in juniperus, leaves all needle-like or all like in rows of 3. The cones are axillary, prostrate, or low shrubs, mostly 3 meters tall. And then we have 1B, leaves mostly scale-like and opposite. Sometimes all shaped leaves present as well, cones, terminal, erect shrubs or trees to 7 meters tall too. And so in this case here, we're going to continue to 2, because we all know Rocky Mountain juniper to be like a small Christmas tree, right? But common juniper, communis, right? It says it's only 3 meters tall, like it lays flat on the ground. So that would be an easy observational character. So communis is not tall enough. We go to 1B, we go to 2. Leaf margins are entire, or leaf margins are denticulate or minutely ciliate. So let's take a look at what that might look like. So hold on one second. And while we're here, we're facing these vocabulary words of entire. What does it mean for the leaf margin to be that, or denticulate, or ciliate? So many choices. Annette, smooth tooth or hairy? That's so much. uh, so in Ackerfield, entire leaf margin is not tooth divided or notched along the margins. And so this is, this is also, you can do this in iNaturalist, just get really up close and personal. And you see how each scale, right? You can see that the edge of each scale is smooth. And so that's how we can arrive at that conclusion. Let me see if I can. 1 seed. All right, so that's as close as we can get to that. But hopefully you can see that from afar, right? It looks real similar, but as we get closer and closer, you can see that there is a lot of going on the margin there. And so it's dentate with very small teeth, and you can see that on the cone potentially easier than you can see it on the leaf. And keep in mind though that cones are modified leaves. Can we all see that now that it's dentate, even though that was pretty hard to see? Obviously a hand lens would be helpful. But I would venture to say like this is even hard to see in an herbarium specimen, because you've got to be looking at those hairs on the mounting paper. And contrast, it's always very helpful. I find it really easier to identify plants first thing in the morning when there's like a little bit of dew. And so that water really helps to like kind of show you those details a little more easily. Okay, so I'm gonna switch gears. I can give you these. I just chose like the best ones from my students' work that I thought you might want to explore. So let's look. Shall we do common yarrow? Can everyone get on board with what that is like? Generally have a mind's eye of common yarrow. Yarrow is a cool one to look at because it's a pretty good one to practice on for a couple of reasons. It is not as sweet. There's not a lot of different kinds of spaghetti. It's not speciose. The same species actually crosses almost all the life zones. All the life zones, actually. I think it's even in the alpine. So let me get there in Ackerfield 1 second. So that's on page 128. Let's go back to briefly the beginning of Asteraceae. So look at 114 for just a sec. And what you're looking at in my screen are all of the observations of common yarrow, Achillea millefolium. It is a worldwide species. Okay, so let's look at Asteraceae. Now, this is one of the most challenging challenging families to get through because it's so speciose. But we have tons of asters in Colorado, and we can practice on one like yarrow that doesn't have a lot of lookalikes. Also not a lot of species in the Achillea genus. So Asteraceae, they're annual, biennial, or perennial herbs or shrubs. The leaves are simple to compound or dissected. The inflorescence ahead, surrounded by one or more series of involucral bracts, phyllaries, which are also called phyllaries, and composed of only disc flowers, discoid heads, ray flowers, and disc flowers, or only ligulate ray flowers. All the choices. The flowers are perfect or imperfect, which is a reference to whether it has male and female parts or just male or female parts. It is sessile on a receptacle. The receptacle is sometimes chafy or naked. The disc flowers are perfect or functionally staminate, actinomorphic, or weakly zygomorphic. And I forgot to mention, as I'm reading, you could just be writing words in the chat, right? We've already come across about 50% of these words need to be defined. Hence the post-it notes. Bottom line is an aster has an interesting compound flower, and we have many of them in Colorado. Let us go now to Achillea, which is on page 127. The reason why I came over here to INAT is because if you filter to plant only and you can filter by person, like you could just look up Jennifer Ackerfield's observations. Her username is Jackerfield. You could look up Southeast Chapter Chair Kurt_Nims. You could look me up, although I do a lot more observing than identifying. Suzanne Dingwell, Suzanne 11. She's a Boulder chapter member, very active in the iNaturalist. But what I'm going to do here is limit it by place. Okay, and so now just by looking at the map, I can tell that like this plant is basically in all the elevations, right? So we can see that it's going from the plains to the continental divide. But one of the neat things about being able to look at many photographs is that hopefully you can find one that gives you a good sense of what you're looking at, right? So let's take a look at this one. Also in iNaturalist, you can really zoom in so that you can count petals, right? Because the number of petals that a species or an specimen has keys you into its family. So let me take a look here. Now, notice that some of these are also called research grade. And what that means is that at least 2/3 of the folks agree that this is what it is. So if I threw in an ID in here and I said randomly that it was Gaillardia, even though I am super wrong, I would reset that clock. I would be part of the masses, and more people would have to say that it's Achillium millefolium to keep it at research grade. So let's take a look. So how many petals does this have? So Jen says 5. I think I would agree. All right, what else can we tell about it, right? What's going on in the middle of these flowers? This is like the key Asteraceae characteristic. Are there 2 kinds of flowers or just 1 kind of flower here? Oh, good one. Yeah. So we have multiple ovaries, and we have ray flowers and disc flowers. And so when we look at this closely, we can see that inside of here, right, every little cluster cup here is actually a complete flower. So this family is to be called compositae. So it's composite of many flowers. So getting into Achillea, so working with the known, right? We're gonna search for the species of known ID. So this is a real short one again. We start with perennial herbs. Leaves are alternate and pinnately divided. So let's see if we can see that. Another thing I would say is it's super helpful if you take more than 1 photograph. And that way you are able to really give the observer as many clues as possible. So I'm gonna look myself up since I'm a chronic multiple photo observer. Watch, this will be one with only 1 photo. All right, so here's some leaves. Can everyone see those leaves? We are told that the leaves are alternate and pinnately dissected or entire. Okay, so let's take a look here. And the only ones that are entire are not native. But can you see how the leaves, they branch off and they branch off again, right? So if you look up pinnately, let's do that for a second. Looking up pinnately, leaflets arranged on both sides of the axis. And you can see that as you look at the vein, there are leaves on both sides. And it's almost fringe-like, right? So now we're gonna go back over here. So the heads are radiate, the involucral bracts imprecate in several series, dry with scarious or haline margins, and a green midrib. Ray flowers are pistillate and fertile, white or occasionally pink or yellow. Pappus, none. Cypselae, flattened parallel to the involucral bracts. All right, phew. So if anyone wants to put in definitions, you can, but I'm gonna keep moving forward because we're running out of time. So our choices were that the leaves were narrow, narrowly lanceolate, and unlobed. Or they are lobed and dissected. And we saw for sure that they are lobed and dissected. So we go to 2, and we have 2A, that the ray flowers are yellow, and they are certainly not yellow. And 2B is ray flowers are white to pinkish, and that's millefolium. And we get to common yarrow. And if you look at the distribution map, it looks a lot like what we saw in iNaturalist. Wide distribution for this species all over the state. And if you were to go backwards, remember that you would go from ray flowers are white to pinkish, right? And then you would back up and say, how'd you get to 2? Oh, we saw that the leaves were lobed and dissected. So, all right, folks. We got through a couple of species. We are running out of time. And I hope this was helpful. Guys, we're a pretty quiet group. We can meet again. You can ask more questions. But ultimately, we will be performing this kind of practice in every restoration committee meeting as we work to really familiarize ourselves with those species on our target species list for the Forest Service project. In terms of your work as a hobby botanist or a professional in the field, keep in mind that visiting the key is helpful because it helps you become a better citizen scientist, right? It helps you to take pictures that convey more information so you can be more sure of your observations. And then finally, you saw some examples in this document that I was sharing with you. That was the work of one of my students. And I appreciated his note-taking skills. But if you are working these species, always take notes, right? You don't want to do this twice on the same species. You only want to do it once.